I would like to share with you an application of the DILIsym software where we will demonstrate how DILIsym can be used to predict and simulate variability and species differences when assessing drug-induced liver injury risk. There are multiple ways that DILIsym can be applied to clinical development, and really it has been designed to assist throughout the pipeline from the earliest in vitro type evaluations to the final clinical trials prior to entering market. For this particular presentation, we will focus in on the two that are highlighted in yellow here, the in vivo, preclinical, and the phase 2, phase 3, phase 4 clinical, in how DILIsym is capable of detecting species differences in evaluating DILI risk, differences that aren't always captured in the classic clinical development safety evaluation procedures. Before we move into the application, I'd like to provide you a brief overview of the DILIsym modeling software. DILIsym includes 4 species: the human, the rat, the mouse, and the dog, and that includes interpatient variability or inter-animal variability. The key cellular processes are all included, as they pertain to drug-induced liver injury, are all included in DILIsym. There's a summary diagram here that kind of describes many of the key mechanistic submodels within DILI, as well as how these submodels interact. Starting from the upper left, you can see that there's a depiction of drug metabolism and distribution, and this enables DILIsym to predict hepatic exposures of parent and or metabolite and how they may interact with the various mechanistic pathways of hepatotoxicity, including reactive metabolite generation, lipotoxicity, reactive oxygen species, mitochondrial dysfunction and toxicity, and intracellular bile acids. These mechanistic pathways can interact with each other as well as with the hepatocyte life cycle to induce apoptosis and or necrosis. There's as well an interaction with the innate immune system within DILIsym to exacerbate and or ameliorate injury. The innate immune system in DILIsym is also responsible for triggering the post-injury increase in hepatocyte proliferation. In addition to all these submodels, there is also a submodel of the various biomarkers of the hepatocyte life cycle. Those include, of course, ALT, AST, and bilirubin, and other more emerging biomarkers. In addition to this quantitative connections of the submodels, we have established the quantitative aspects of within and between the submodels by using exemplar compounds, particularly those focused on specific or more than one DILI mechanism. And DILIsym is what we describe as a compartment-based mathematical model that utilizes ordinary differential equations. Also, we preserve mass balance even in the loss of hepatocytes by using what we call a form-to-function connection, wherein loss of hepatocytes kind of requires that drug and circulating metabolites be taken up by only the remaining viable hepatocytes. So on to the example. So Troglitazone was a treatment for type 2 diabetes that reduces hepatic and peripheral insulin resistance. It's a was first in class of the thiazolidine diones, which means it's a PPAR gamma agonist. However, there was clinical hepatotoxicity observed in patients that wasn't detected in preclinical rat or dog studies. And the hepatotoxicity that was observed in the patients was relatively low, at only about 2% of the patients developed ALT elevations greater than 3 times the upper limit in the clinical trials. Nevertheless, it was withdrawn from market due to this idiosyncratic hepatotoxicity. And there have been a number of studies over the years since its withdrawal from the market, and hence many possible mechanisms may contribute to the hepatotoxicity. The focus of these simulations, however, is on the bile acid mediated toxicity pathways. Let me summarize those for you here. And so the bile acids are taken up from the circulation via NTCP upon entry into the hepatocyte. They can be exit the hepatocyte via the MRP3/4 on the basolateral side, or they can exit on the canalicular side via the bile salt export pump, or BSEP. Troglitazone, upon entering the cell, is primarily metabolized into troglitazone sulfate, its main metabolite. Both compounds, the parent and the metabolite, inhibit BSEP and MRP3, which causes the accumulation of bile acids and can lead to hepatotoxicity. Troglitazone in the circulation also inhibits NTCP, which to some extent should mitigate the accumulation of bile acids by reducing the amount of bile acids that enter the hepatocytes via the enterohepatic recirculation. So with that in mind, we simulated the inhibitory properties of troglitazone, and in our SimPops, which account for the inter-individual variability between patients. And you can see here, in this figure, there are a few observations that I think are worth pointing out. First off, there's something of a dose-response effect, where at 200 milligrams of daily simulated troglitazone for 6 months, there's very little increases. Very few of the patients have increases in ALT. However, as the doses increase to 400 and 600 milligrams, an increasing proportion of simulated patients do have clinically relevant increases in ALT. So that's one thing to point out. The other thing to point out is the relatively low frequency of the increases in ALT. Even at the highest 600 milligram dose, the proportion of patients with ALT is still only about 5% in the predictions with these SimPops. Thus, there is very clearly interpatient variability, and by the way, the predicted frequency is consistent with what was reported from the clinical trial data. So if we take this forward and compare now the simulated response in humans, which again was consistent with the clinical data, to the predicted responses in the rats, what you see is on the right, for the rats, there's very little, virtually zero, predicted increase in ALT. Particularly there's definitely zero beyond the 3 times the upper limit of normal. Whereas again, in the humans, there was 2 to 5% predicted incidence of ALT increases. So there's a very clear species difference between the rat and the human. And in fact, as I mentioned previously, the rat did not, or rather in the preclinical rat studies, there were no reports whatsoever of drug-induced liver injury with troglitazone treatment. So again, the species difference that was observed in the preclinical or the clinical development approach was also captured with these simulations. It's worth pointing out that had the simulations been done in advance of the clinical trials, that this risk would have been highlighted and the clinical trials might have been designed differently. So another thing to point out here is on the mechanistic side, DILIsym has the ability to ask some key what-if sort of questions that enable the user to gather even more information about the underpinnings for a given simulation. So we've already described the fact that there is interpatient individuality in the responses, some differences there. And so the question is, how much does each of those specific bile acid transporters contribute to this interpatient variability and the predicted response? So again, the overall incidence predicted when the inhibition of all 3 transporters is active is about 3.6% in this SimPops. If we remove the BSEP inhibition, it is reduced tenfold the incidence, the predicted ALT increases. Whereas if we render MRP4 inactive, again, it's virtually no ALT increases. If we render NTCP inactive, then actually there's an increase in the proportion of ALT incidence. So again, I think what we learn here is that individuals who come in with perhaps low BSEP to begin with, or MRP4, or perhaps both, those are the ones that are likely to be the most sensitive to the inhibitory effects of troglitazone, and helps us understand some of those interpatient differences. So to summarize, DILIsym successfully simulated the species differences in DILI that were observed with troglitazone treatment. This has been written up and published in a nice paper by Kyung-hee Yang. And again, this was due in large part to the types of bile acids that are present primarily in rats and humans, and how they're different, and the hepatotoxic potential for each is different. Also, I should point out that within the patients alone and the inter-individual variability, that differential response was captured in the simulations. In addition, the simulations provided some mechanistic insight into the individual contributions from the specific bile acid transporters and how they were inhibited to the overall predicted DILI. We're happy to report that this publication was supported as well by a comment from members of the FDA Office of Clinical Pharmacology, and this comment was published in the same issue as our paper. And the quote I'd like to read for you is that we look forward to future efforts to apply this model for prediction of hepatotoxicity that has not been clinically observed. So certainly we in the DILIsym team echo these sentiments, and we look forward to applying it in that way.